Thanks, Jody and Krishna, for the introduction. Way we're going to do this is I'll start and then John will take over partway through. The talk is mixed between kind of introductions, which use artificial made-up data, and then a case study that John will cover. Okay, let's get going. I think we should start out talking about what synchrony is, just for those who aren't familiar. The word is used in a lot of different ways, but what we will mean today when we talk about synchrony is also sometimes called spatial synchrony or population or metapopulation synchrony. And it's essentially when populations of the same species in different places fluctuate in related or correlated ways. Other types of synchrony include community synchrony and phenological synchrony, but we're talking today about spatial synchrony. So here's two examples, these two panels. The one on the left is more synchronous time series. The one on the right is less synchronous or asynchronous time series. In both of these cases, what we're imagining is different populations in different places of the same species, which have been measured through time. So why should one study synchrony? Well, probably the biggest and best reason is that it's just a ubiquitous phenomenon that has turned out to play a role in many other topics in ecology. One of those roles, other than it being ubiquitous, I should say sort of one of its applications, it has importance for aggregates. So suppose your populations in this example in the upper left are from different places and you're adding them all up across space to get a total population, or let's say you're taking the average across space to get an average across this larger region. If you sum or average these time series, you'll get a highly variable aggregate. Whereas if you sum or average these time series, you'll get a much more temporally stable aggregate. So, and basically the only difference between these two sets of time series is the degree to which they're synchronous with each other. Other statistical features of these data have been retained across these two scenarios. So what you can see impacting the temporal stability of the aggregate here is the synchrony. So causes of synchrony, it's been known for decades theoretically that, well, more than theoretically, but originally theoretically, that synchrony, that there are some causes of synchrony. Synchrony of environmental drivers, like it might not surprise you to learn that if you've got an environmental factor that's influencing all of your populations across a large area, and if that environmental factor is itself synchronous across that large area, then it may tend to produce synchrony in the populations. That's called the Moran effect. Dispersal is another, if your populations are actually exchanging some dispersers. In some cases, just a small amount of dispersal can be enough to bring the populations into synchrony with each other, even if there aren't Moran effects operating. And a third cause is if your species that you're interested in, your taxon that you're interested in, is maybe not influenced, maybe not engaging in dispersal between your populations, and maybe not influenced by an environmental driver, which is synchronous, but it is influenced by either a synchronous or a mobile predator, or any other species that it may interact with, maybe not a predator, but maybe a prey or a competitor or something, then that has the possibility of bringing your focal taxon into synchrony across the space. So historically, it was considered hard to distinguish these causes in any given field system. But lots of techniques have sprung up over the last, let's say, 20-ish years that have made that kind of inference much more straightforward. So the paradigm has expanded. So I think now researchers by and large think that if you have enough data and you maybe get a little bit lucky, but mostly you just need enough data, you can probably infer the causes of synchrony in your system. So the goal today is to introduce this package wsyn and what can be done with it. Two main things I think we should say that are what wsyn specializes in. It facilitates a time and a time scale specific analysis of synchrony. I'll go into in a bit what that is. And it facilitates some of those inferences of environmental causes of synchrony, namely Moran effects that I mentioned just a minute ago. So before we get into that, though, I want to give a little nod to some history We can't, of course, explore all of the history and do a complete review in the context of this talk, but I want to sort of mention a few papers just as a way of kind of respecting what came before. Timescale specific approaches, which are what wsyn are centered on, have been developed through time, certainly not starting with wsyn. So has consideration of changes through time in synchrony, and so have efforts to infer causes of synchrony. So all of these themes have a lot of history. We can't review it all, but we'll mention a few things, partly for entertainment value and partly just to show that there's this history. So before we do that, I want to just say what timescale specific approach is. What is a timescale specific approach to synchrony? Well, synchrony is relationships between time series, and what's been realized is that you can have relationships between time series which are different on some time scales than what they are on other time scales. And these are just a couple of complete, obviously made up examples. You can see both in this top scenario here, we've got two signals, two time series, both of which are the superposition of two sine waves, a slow one and a fast one. And you can see if you match up the peaks with the troughs in the top time series match the peaks in the bottom time series on the fast time scale. So on the fast time scale, the oscillations in these two time series are actually anti-synchronous with each other. But on the slow time scale, you can see that the fluctuations are aligned. So on the slow time scale, the two time series are actually synchronous with each other. So we've got opposite patterns of synchrony and anti-synchrony on two different time scales here. The example on the bottom is analogous, but I've flipped it. I've made it so that the fast time scale oscillations are now synchronous, as you can see, because the small peaks align with each other, but the slow oscillations are anti-synchronous with each other. So that's just to show that, synchrony, it's got to have some kind of, it may potentially have in real data as well, some kind of decomposition by time scale. So here's an early influential example of a timescale specific approach. This is really a great paper to read. It's well written in many ways, and it's fascinating. They studied measles dynamics in the UK. Measles had a biennial outbreak dynamic in the pre-vaccine era. And by studying synchrony at that specific time scale, the authors were able to demonstrate traveling waves and changing synchrony through time as vaccination set in. So it was key to their approach. Their approach was wavelet based, and I'm going to use wavelets later. And it was timescale specific in the sense that it focused on the frequency at which this biennial peak occurred, this biennial outbreak periodicity occurred. So here's a wavelet. This is one of their figures. Here's a wavelet reconstruction of annual fluctuations in 3 cities in the UK, London, Cambridge, and Norwich. And you can see this is a wavelet reconstruction of the biennial fluctuation. You can see that Norwich, which is the blue one, is out of phase with the other two. And then as the vaccine era sets in, it comes into phase. I also wanted to give one early example of changes in synchrony through time. The Grenfell study, this study shows changes. You can see that here as the synchrony changes. But here's an even earlier one. Steen in 1990 showed changing synchrony in an amazing 79-year time series of small rodents in Norway. And here's the way that they pictured that change in synchrony. It's different from Grenfell. It's different from what we're going to do, but it still shows the change in synchrony. So a 1 here encodes lots of small rodents, and a 0 encodes not so many small rodents. You can see time across the top here. And if you trace across here, you can see there's periodic fluctuations which seem pretty synchronous across these 4 different portions of Norway until you get up to the early 1900s here, where there's a disruption to that pattern. And then the synchrony reasserts itself in the later portions of the time series. So that's a change in synchrony, even if I'm not going to go into potential causes for it. Here's an early example of inference of causes of synchrony. I like this example because sometimes when we say inference, we think formal statistical inference, but inference can be considered more broadly than that. So this is just an inference from the scenario on the ground, from the way that this system was known to work. So this is essentially a special case where two of the three causes of synchrony that I mentioned on an earlier slide were impossible. So they studied populations of sheep on two islands in the St. Kilda archipelago in the North Atlantic. So this is what we're looking at here. We've got, say, a population of sheep on this island here, and another population of sheep on some other island 5 kilometers away. There's no reasonable way that sheep would be swimming across 5 kilometers of North Atlantic Ocean, and so dispersal was not possible, and the sheep were known to have no predators. So that means any observation of synchrony, which there was between the two islands, was going to be Moran effects. So having done just a nod towards some of the earlier work on some of these themes that wsyn overlaps with, I want to restate the goal. So the field has matured through time and has adopted increasingly of a time and time scale specific approaches to synchrony, how they change and how they're structured in time scale. And inferences of causes of synchrony are now fairly common. One of the frameworks that's grown up to support those kinds of analyses is implemented in wsyn. And so we want to introduce it and what you get from it. And the viewpoint throughout is going to be operational in the sense that we'll show something about how to introduce these tools. We'll show something about how you interpret the results you get. The math is going to be, there will be some math, but it'll be pretty minimal. But there's detailed references if you want to dig into the math. And so we're going to first introduce tools in wsyn using made-up data. And the reason why we feel like that's a good way to do it is because if you make up the data in a certain way where you know to expect certain patterns, and then you get those patterns from the tool, that the tool reveals those patterns, then you can tell that the tool is working. You can try to understand, well, you can understand how it's working operationally. So we're going to use made-up data, and then we're going to use real data from a kelp system, and Jon will talk about that part. So as I understand is normal for this seminar, all codes for worked examples are on the GitHub, and a lot of those codes are also available through the package vignette and were released with the publication that Jon will talk about. But we've got simplified versions on the GitHub. Okay, so the first tool that everything else is built on and based on in wsyn is the Wavelet transform, which is quite a general tool used in many other scenarios, but I think it needs to be introduced for people that aren't familiar. So we're going to do that. So in the context of this example that I'm doing right now, data just consists of one time series. So given a time series, the Wavelet transform, what is it? It's a function of sigma, which is time scale, and t, which is time. And for each sigma and t, it's a complex number that has two pieces of information in it. The magnitude of that complex number is the strength of the oscillation in the time series at time t and at the time scale sigma. I'll show pictures of this later. And the phase, the complex argument or phase of the Wavelet transform at sigma and t is the point of the oscillation that you're at that time. So the Wavelet transform decomposes variation by time scale and tells you how that decomposition changes through time. If you're familiar with Fourier analysis, you can think of Wavelet analyses as being essentially more or less time-windowed Fourier transform, but done in a statistically better way. So let's see this work on some fake data. So let's make up some data first, and then we're going to take a Wavelet transform of it. So we're going to make one time series, which is a sine wave of period 15 that then switches periodicity to period 8, right in the middle, with some white noise added on top of it. So here's the code that makes that. Here, this line is the first part of the time series having its period 15 signal, and then it later changes to period 8. And then here's the white noise, and then we add them all up. So we're going to apply this WT So all my functions that we're applying are from wsyn, so I always put the wsyn there in front just to remind you. We have this clean.dat function, which comes in handy, but we can sort of skip the details of that right now. And then we and then we use this wt function, which is the wavelet transform. That'll return you an S3 class, an object from an S3 class, which is for holding the results of wavelet transforms in the package. There's a bunch of other arguments to WT, which specify details about what version of the wavelet transform we're going to use, but default values for, these are Morley wavelet transforms, by the way. Default values for other WT arguments are generally good enough for data exploration. And if you want to dig into the specific values of those, you can see the vignette. So having taken a wavelet transform of our data and stored it in this object wtres, we can now use a function called plotmag, which is going to plot the magnitude of the wavelet transform as a function of time and time scale. So here's what you get here. Time scale on this axis, time on this axis. Color gives the magnitude of the wavelet transform, and the magnitude of the wavelet transform shows you how much oscillation is in the time series at a given time scale at a given time. And now you can see what we built into the data, and this is where I think the strategy of constructing data and then subjecting it to an analysis can be useful, because you know how we built the data, you know what should be there, and now we can verify whether it is or not. So we know we built in a time series that had oscillations at period 15, and then the oscillation switched to period 8, and you can see that clearly in the magnitude of the wavelet transform. The phase of the wavelet transform, I'm just showing it for completeness. You can see basically there are oscillations taking place in the wavelet at each of different time scales. There are oscillations occurring at the time scale, at about this time scale that's indicated on the y-axis. We're not going to use this. I just wanted to show it so you can be reminded that there is also phase information. We are going to use phases, but we're not going to use this plot. Okay, so the power spectrum, if you're familiar with just classical Fourier analysis, the power spectrum wouldn't show this change. It would just show two peaks at time scales 8 and 15. It doesn't have any time resolution. It just is frequency resolution or time scale resolution. So what the wavelet offers you is both time and time scale resolution of what's going on. So not only does the wavelet transform show the overall variability of the time series decomposed by time scale, it also shows how that changes through time. So all of this, wavelet transforms have been around for quite some time. This is just an introduction because we're going to build on it. Okay, so we haven't even talked about synchrony yet. That first example, the data was just one time series. You need multiple time series to talk about synchrony. So that's what we're going to do now. How do we use wavelet transforms to display how synchrony depends on time and time scale? Excuse me. So data will now, for this next portion of the talk, consist of multiple time series from different locations. So that's the setup that we assumed from the beginning when we talk about synchrony. So now we've got these time series. I'll denote them by x sub n as a function of t, where little n goes from 1 to big N. Those are your different locations where somebody has spent years gathering data. Thank you. And wavelet transforms, we're going to denote like this. So we used to just have one single wavelet transforming. It was a function of time scale sigma and time t, but now we have n of them, capital N of them, indexed by the little n here. So we're going to define something which is called the wavelet phasor mean field. This has been used longer in biophysics than in ecology, but it's getting used more and more in ecology now. And it's kind of simple mathematically. It's basically just you're going to take the wavelet transform, you're going to divide by its complex magnitude so that you just get a unit magnitude version of the thing. And then you're going to average those across all your locations. Now, one word that's sometimes used for this unit magnitude complex number is phasor. So I'll go ahead and use that word. We're going to convert our wavelet transform into a wavelet phasors. which just have the phase information. And if we plot this as a function of time and time scale, it's going to give a pretty decent depiction of how synchrony in these time series depends on time and time scale. How does that work? So this is the kind of plot that you get from it. It's going to have time scale or frequency on one axis and time on the other axis, and color gives the intensity of a kind of synchrony between the time series. So you'll be able to see, for instance, that like this red blob, which occurs about once every, corresponds to a time scale of about 10 years and occurred in, let's say, the mid-80s, that the time series were a lot more synchronous then and a lot less synchronous at this other combination of time and time scales. So how does that work? So here's the complex plane, and these are each of our phasors from different locations. And the idea is if the oscillation at the given time and time scale is at the same place in its oscillation, in its phase, if it has the same phase across all your locations, then all these complex phasors will point in about the same direction. And when you average them up as complex numbers, you're going to get a big complex number. Versus if they're all in different phases, in other words, the different signals in different places are all at different portions of their oscillation, different times in their oscillation, then those phasors are going to add up to not much because they're just going to wander around randomly in the complex plane. And you can see that done for the actual data that underlie this example. The phasors for this green triangle here were all pointed in about this direction. And so they add up to a big number, and their average is a big number, versus in this point, they add up to not much because they're all kind of disagreeing with each other. So this wavelet phasor mean field measures the phase synchrony of your different signals, of your different populations, as a function of time and time scale. Let's see that in fake data. As is our general setup, we make fake data, we try out the tools, we see how they work. So we're going to make some fake data for this case too. Several time series, because that's the context. We're going to make up these time series that they're going to all have one underlying signal, which is used in all the time series. So it will contribute synchrony to all the time series. And that signal is going to switch from period 10 to period 5 halfway through. And then we're going to add some randomly phase shifted period 3 sine waves. So all these signals, all these populations in different locations, they're all going to, in addition to this one common signal, they're all going to have a period 3 sine wave, but I'm going to randomly phase shift all those sine waves independently in each of the locations. And then I'm going to add some white noise on top of that. So here's the code to do that. Here's the underlying signal, which is common across everything. And then I'm going to go through in a for loop here. And for each of my time series, which are stored in this matrix, I'm going to put in a period 3 sine wave. Here's the period 3. And I'm going to randomly phase shift it. That's what this is. And then I'm going to add some white noise to it. So, and then I'm going to add them all up. This is the common signal plus the randomly phase shifted period 3 noise, or period 3 oscillation, plus the white noise makes the counter row of my data matrix. So that's the fake data. Let's look at it visually. One thing that I really like to point out in examples like this is the classic methods of looking at synchrony. Is you can't see it visually. Like there should be some synchrony built into these data because we put it there, right? We had this underlying sine wave, which was present in all the data sets, in all the time series. So we know there's some synchrony there. But if you just plot the data, you can't really see it visually. And if you just plot pairwise correlation coefficients between all the different pairs of time series, you can't really see it there either. Like they're spread out all over the show. Maybe there's a slight bias towards positive correlations, but not enough to really think much of. Now, if we apply this wavelet phasor mean field, that's supposed to give you a time and time scale specific picture of synchrony. So it should reveal what we built into the data. So this WPMF function gives you another type of S3 object, which is for the results of wavelet phasor mean field calculations. And again, there's a bunch of default values which you don't have to worry about, but you can see the vignette. So once you compute a wavelet phase or mean field and store it in this object WPMF res, you can use a plot mag. This is an overloaded function, so it'll produce a different result for depending on what the class is of what you're putting in here. And this is the resulting wavelet phase or mean field. I'm asking for a significance threshold of 0.95, and you can see that there is the synchrony that we couldn't see here. We can now see it in the wavelet phase or mean field, and it is what we know that we built in, because we knew that the synchrony was caused by that one underlying signal, which I built into all of the signals, all the populations, started out at 10, then went to 5. So now we can detect this time and timescale specific synchrony that was built into the data. We couldn't test it here, essentially because the synchrony that we knew existed at certain timescales and also changed through time was obscured using, if you're just using Pearson correlation, it was obscured by the asynchrony at other timescales and the fact that there was this change through time. So there's a significance built in. I've set, I've put these contours which correspond to significance, so I need to say what I mean by that. This is significance as compared to a null hypothesis of random independent phasors. So if you compare the wavelet phasor mean field, the sum of phasors that you actually get, to what you might expect to get if all those phasors were just randomly on the complex unit circle, then that's the comparison that leads to this null hypothesis. And so what we're saying is, when we're saying there's significant phase synchrony, is that these phasors are significantly more aligned than random. And as always, you need to keep in mind multiple testing. I'm showing a lot of tests here effectively, and you can see that some of them, there are some type 1 errors on here. All these blobs down here, this blob, and some of these blobs are going to be type 1 errors, and that's just, everybody is aware that that's a price you pay by doing statistics. Okay, we also use another related tool, which I won't delve into. It's called the wavelet mean field. It's similar. You can, with some differences, you can find it in wsyn with this WMF function. You can interpret it, all I'll say about it now is you can interpret it more or less in a similar way to WPMF, and you can see the vignette for details. So now what do we have here? This example shows some benefits to a timescale specific approach over classic correlation-based methods. Basically, the correlation methods didn't show the synchrony that we knew was there because we built it into the data, because those methods conflate timescales and average across time. Synchrony on some timescales was obscured by asynchrony on others. And if you use a tool like WPMF or WMF or other tools which are out there which do something similar, you can now see synchrony on its timescale structure. It turns out, you wouldn't know it from these examples, but it turns out that timescale structure is quite common in synchrony, and changes in time are also common in synchrony, and are a consequence of climate change. Okay, so now I want to start to explore causes of synchrony, because that's where I promised we would go from the beginning. We know that one important cause of synchrony is the Moran effect. That was one on the list of three theoretical causes at the beginning. So how do we start to explore that from a time and timescale specific viewpoint? To do this, we need to understand relationships between two sets of time series measured in the same places, where usually one of these is a set of environmental time series, like temperature measured, let's say spring temperature measured every year in all the same places where you census your population. And the other one is biological, that's censusing your population. So at first we started with a data which was one time series when we looked at the wavelet transform. Then we used data which was multiple time series when we looked at the wavelet phasor mean field. And now that we want to think about causes of synchrony, environmental causes of synchrony, we need two sets of time series measured in the same times and same places. because we need to have our environmental data as well as our biological data if we want to think about whether one might have caused synchrony in the other. So our approach here is based on something called the wavelet coherence. So that's what this next part of the talk is to introduce wavelet coherence. I want to do it by analogy to start with, because I think the analogy is pretty cool. Biology, as you all are aware, it has all kinds of irregularities and randomness, and it's different from physics in that respect, right? It's got all kinds of weird stuff going on all the time. So what our method does is it's not our method. What wavelet coherence does, we use it a lot, so sometimes we start to call it our method. We capitalize on the very irregularity of biological time series to make some of these inferences. So let me try to explain what I mean by that. If we imagine two oscillators going through time, and they might be related to each other, we want to quantify the consistency of the phase relationship between those two signals through time and across space if we have multiple signals. And we want to compare it to what would be expected if the signals were unrelated to each other, but had the same degree of phase drift, the same degree of irregularity in their oscillations across space and time. So let me give an analogy here. Suppose we call each other every day, you and me, and I have a clock in my office and you have a clock in your office. And every time we call each other, for some reason, I tell you what time my clock says and you tell me what time your clock says. And it's always, we notice it's always 2 minutes different. Every day, whenever we call each other, whatever time of the day, our two clocks are always 2 minutes different from each other. That doesn't mean anything, right? Because clocks are extremely regular oscillators. If they're good clocks, they're very regular oscillators. So if by chance our two clocks started out 2 minutes out of phase with each other, they should, if they're good clocks, remain 2 minutes out of phase with each other for a long time. But let's suppose your clock is actually a terrible clock, and it's always gaining and losing time all the time, randomly. And my clock is also a terrible clock. It's always gaining and losing time randomly. So now we've shifted to biological oscillators. That's what biological oscillators do. There's always a degree of randomness and phase slippage going on. So suppose even if our two clocks are bad in that way, still every time we call each other, we still notice that your clock is always 2 minutes ahead of my clock. Well, that is actually really good evidence that there's some relationship between the clocks. And we don't know whether maybe your clock is telling my clock what to do, or my clock is telling your clock what to do, or maybe they're mutually related, or maybe there's some outside force affecting both clocks. But because they're very irregular, but they still maintain a consistent phase difference, that provides good evidence of a relationship. And that's basically what the phase coherence quantifies. So wsyn has a function which does a spatialized version of this classic wavelet coherence tool to detect relationships between variables in a timescale specific way along the lines of that analogy that I just said. So if these are the two wavelet transforms, they've been normalized, but we can sweep that under the rug. Then we can compute this quantity, which is one of the transforms times the complex conjugate of the other one. And this is the phase difference. This is like the 2 minute difference between your clock and my clock. This tells you how distinct the two phases are of the two things. And then we're going to average that phase difference across all the times for which we have data and all the locations for which we have data. And so basically when we average this phase difference, this product here, if it adds up to a big number, then that is because the phase difference between these two wavelet transforms is consistently maintained through time and across space. If it adds up to a small number, then that phase difference is not consistently maintained. So, and what's important is you compare that quantity against the same thing calculated for what we call surrogate data sets, which is a kind of bootstrapped data set. In this case, we use bootstrap data sets that maintain the temporal autocorrelation, basically the spectral characteristics of the signals. So what we are asking is this phase difference more consistent than what you would expect if compared to these surrogates, which are unrelated to each other, but maintain the same temporal autocorrelation signature, the same spectral characteristics as the original data. So let's see that in the fake data. Let's make some fake data and then test the tool on it. So the fake data are getting more complicated than they were in previous examples, but they're still reasonably simple. So we're going to now, consistent with our new context, have an environmental driver and a biological variable, both measured at 11 locations measured through time. So the environmental variable is going to be the sum of a weak common signal at period 10, which is present in all locations, a strong common signal of period 3, also present in all locations, and white noise. So here's the code for making that. Here's the weak period 10 signal, weak because it's amplitude 1. Here's the strong period 3 signal, strong because it's amplitude 5. And then we're just putting both of those in all locations, and we're adding a bunch of white noise. Now, now we have to make up fake biological data. The population time series are going to be driven by the environmental variable in each location. You know, each biological variable, each population in each location is driven by the environmental variable in that same location, using a 3-step moving average. And then there's going to be white noise added on top. So you can see that here. We're just going through a for loop here. And for each, this is going to hold our biological data. And then we're going to take the mean of the the current environment and the 2 previous environments, plus some white noise, is going to give you your population in each location. Okay, so why did I make it a 3-step moving average? Well, that interacts with this strong noise here. A periodic signal of period 3, if it passes through a moving average of 3 time steps, is going to be averaged away. So this portion of our environmental signal should not pass through to affect the biology because it gets averaged, but this portion, it should pass through. Okay, so the question now is, can you see the relationship here between the environmental and biological data just using ordinary correlation? Not really. So this is a histogram of the location-specific correlation coefficients between the biology and the environment. Basically, you can't really see what's going on because the driving that is taking place is just swamped by the other components of what's going on, the white noise and the moving average signal not passing through. But now we can use this tool in wsyn, which implements the spatial wavelet coherence. It's called COH for coherence. And basically you call it, most of this I'm going to kind of ignore or briefly say something about it, but you can see I'm passing in the 2 data sets. The environmental variable, the biological variable. You also have to give it the times. And there's different ways that you can measure significance. And it's based on bootstrapping. So this is the number of randomizations to use. And we'll forget about those other 2. And again, you get another S3 class, which is defined for holding coherence results. And you can see the vignette tells you exactly how to use this tool. Now, what do you get from it? You get a plot that looks something like this. You can get various kinds of output from it, but this is maybe the first thing that you get. So it's coherence on the y-axis, time scale on the x-axis. The red line is the coherence, and the black lines are confidence levels, thresholds, based on surrogates at 95 and 99%. So you can see, as is consistent with the way we built the data, the period 3 environmental fluctuation, which was very strong, didn't make it through and didn't end up establishing a pattern in the population which would yield a coherence with the environment, because it's a period 3 oscillation, and we know that the way that we set up the environment drives the populations with a moving average of length 3. So that's why you get a low and non-significant coherence at timescales around 3, but you get a high and significant coherence at these longer timescales, which correspond to that influence of that much weaker period 10 signal that we built into the biology. So even though the period 3 environmental oscillation was much stronger than the period 10, it doesn't influence the populations because of the moving average way that these two things are related to each other. And we can see that the population-environment relationship depends on time scale. You have to account for that if you want to detect it. As we sort of sketched here, you couldn't really see it with just a correlation because it only happens on some time scales. So this shows the relationship. It shows that it's a timescale specific relationship, and this is also going to influence the potential for Moran effects. There's some other ways of plotting the same result. This just shows the rank of the coherence compared to the bootstrapped coherences. And when that value goes high, higher than a significance threshold, which is the dashed line here, then you can, for those timescales, you can consider the relationship between them to be significant. There's significant wavelet coherence there. You can also, wsyn will also allow you to add p-values for particular bands. And these are an appropriately kind of average p-value across the band, which can help mitigate the multiple testing problems. So you can see this band is the 8 to 12 band, and it's highly significant. This band is the 2 to 4 band, and it's not significant because of the moving average. I think in the interests of time, what I'll do is I'll say there are some different, you could talk a while about what are the different surrogates, what's the bootstrapping approach here. I think I'll just leave it at saying basically what we do is we construct bootstrap time series which have the same spatial autocorrelation and the same temporal autocorrelation properties as the original data, but we had two sets of original data. If you produce surrogates of one of them, then the surrogates are now unrelated to the second set, even though they maintain the spatial and temporal autocorrelation of the original data. Okay, so advantages here of this timescale specific approach. Time series can be related to each other on some timescales and not on others. This should, by the way, just thinking biologically, be pretty common if it's common that populations are, let's say, influenced by a moving average of environmental conditions. If I'm, let's say, a bison, I don't really care what the temperature is today or yesterday. What matters is longer term averages, because my physiology is such that I can average over rapid temperature fluctuations. So moving averages effects in biology are probably pretty common, and that was built into the example that I did. Coherence can address some of that. It turns out that timescale specific relationships are common in biology, and the examples that we've explored over the past 10 years or so. Okay, so how do we get from here to saying something about environmental causes of synchrony? I said that those tools were going to be built on wavelet coherence, and they are. In the interest of time, I'm going to get more sketchy than I was before, but I am going to kind of outline what can be done now that we have introduced the main statistical ideas here. So how does coherence relate to possible causative relationships? And how do we attribute synchrony to particular causes? And what about interactions between causes of synchrony? So it is admittedly a fraught topic whenever you start trying to make causal statements. There are, of course, several statistical frameworks for doing that. And in the context of our particular context, wavelet coherence and related tools give another such framework. It relies on a particular context, though. And that context is when we have an environmental driver and a biological variable. Well, we don't know if it's a driver, but we have an environmental variable and a biological variable. It's not plausible on the timescales and the spatial extents that we're thinking of to imagine the causation going the other way, and that's a crucial part of the argument. So if A is an environmental variable and B is a population variable, and A and B are highly significantly coherent on a certain timescale band, then logically you have to have one of the following things. A is driving B on that band, possibly indirectly, or B is driving A on that band, but that's very unlikely because B is the biological variable and A is the environmental variable, or they're mutually influencing each other, again unlikely, or some third variable C is driving both. That could well be the case, or driving B and mutually related to A. But in that case, C is probably another environmental variable, so you've still demonstrated Moran effects. So what we have is if A is an environmental variable and B is a population variable, and A and B are highly significantly coherent on a certain timescale bands, and also if A is synchronous, then we can reasonably claim to have identified a Moran effect. It could be coming from another weather variable related to A. And then can we quantify how much synchrony in B is explained by synchrony in A? And this is where wsyn comes in. I'm not going to fully demo these tools. I'm just going to state that they exist and they're available in wsyn, and I'll tell you what they can compute. They're based on some theorems which are published in these locations, and they're described, and there's more examples similar to the ones I've done in Spirit in the vignette. They all rely on this function, the WLM function, which stands for Wavelet Linear Models, which is in wsyn. What can these tools do? You can investigate which environmental variables most likely helped cause synchrony in a set of populations on any given timescale band. I mean, you're limited by your data. If you don't have data on some environmental variables, then you can't investigate those ones. You can quantify the fraction of synchrony explained by each of these factors, and you can quantify the fraction of observed synchrony explained by pairwise interactions between them. And that's all I want to say. John is going to delve more into WLM as well as putting some ecological teeth on these tools.
Cool. Well, thanks to Dan for providing that background. I want to build on that by talking about an empirical example that's focused on the dynamics of resource subsidies in the form of kelp wrack. And that's these sort of brown mounds of detritus that you see washed up on the beach in this photo. And if you're following along with the code from here forward, you'll want to look at the example _kelp_wrack file. Right. And so basically, what we want to do in this section is to hopefully make what you learned a little bit more concrete through application to a real system, and also to kind of aid folks in thinking about how they might apply these tools to their own interests by discussing some of the challenges that are posed by real-world data. So a little bit of background here. Giant kelp is a foundation species of rocky reef ecosystems. We found it to be a really awesome system for studying synchrony because it grows really rapidly. It forms a canopy on the surface of the ocean that can be detected from satellite imagery. And this gives us population time series at unusually high spatial and temporal resolutions over large areas. And then in the context of this study, kelp also provide important resource subsidy to sandy beaches in the form of its detritus or wrack. And this supports a complex food web that really wouldn't exist otherwise, since beach ecosystems are pretty low in situ primary production. And this example is taken from a paper that came out earlier this year. I'm using this data, adapting some code from that paper to provide this demonstration. Unfortunately, I don't have time to kind of cover everything we cover in this paper, but definitely love for folks to check that out if you're interested. So in this study, the punchline is really that across these 5 beaches that are sampled by the Santa Barbara Coastal LTR site, we see that the kelp forest and the beach are coupled through processes where kelp is produced offshore on reefs, It's dislodged by waves and transported to the beach. And then beach morphology affects whether it can receive and retain wrack, and then wrack affects the beach community. And that's exemplified in this study by the local abundances of shorebirds that forage in the wrack, feeding on invertebrates. And crucially, this occurs synchronously across all of these sites. And in case it's not already clear, in this example, we're kind of expanding the focus of spatial synchrony just a bit from the examples that we started with, talking about resource subsidies as our focal variable, and not population abundances. And one thing I hope that this kind of underscores is that the tools that we've developed, while they were designed around population spatial synchrony, they can also be extended to other types of synchrony that folks might be interested in. If you're thinking about communities, other sorts of variables, we've worked on phenological variables. We've also worked on biogeochemical variables. And all of these things can kind of fall under the umbrella of this package. Here's where our data come from. Big shout out to our collaborators, Jenny Dugan and Dave Hubbard and Kyle Emery and others who have generated the beach data. This is a field sampling program that a lot of people have contributed to. And then Tom Bell has led the development of the kelp and oceanographic data. Like you've started to get a taste of, we have some conventions for data structure and completeness. And if you're following along, I've organized the data into site by time matrices. And since we require complete regularly spaced time series, I've filled in gaps with the median value for that site and that month. And that brings us to some recommendations for filling gaps. We often need to do this. It's common for data to have missing values in it for a whole host of reasons. But just like any other analysis, the way that we fill those missing values, the way we deal with those missing values can be important. So here are some thoughts about how to do it responsibly. And this is a little bit unique for synchrony studies. We generally want to really avoid borrowing information from neighboring sites, like we might say with kriging or other spatial interpolation methods, or different kinds of spatiotemporal models. And this is because when we borrow information from neighboring sites, by construction of these approaches, we're going to tend to inflate the amount of synchrony that's measured, and that's going to muddy the waters of how much of what we're analyzing is the data itself versus the interpolation method. And then we also want to be cautious with temporal structures. That's what a large part of the methods that we're using are leveraging for inference. So we want to make choices that minimize assumptions about what's happening during unmeasured periods while preserving important features. These are usually things that have well-defined periods and consistent amplitudes, like seasonal and diurnal cycles, depending on the data resolution. And again, really want to minimize the assumptions that we're making about what's happening during unobserved periods. So for us, a typical practice is to fill gaps in annual time series with the median value for that site. For monthly time series, like in this example, we will fill our gaps with medians for that site and that month. And I haven't analyzed that many daily or sub-daily time series using these tools, but when I have, I've actually used linear interpolation for short gaps. And lastly, it's important to note that there are some limits to what you can responsibly fill. We've generally avoided analyzing time series with more than around 20% missing values. We've also generally avoided analyzing time series with many or all locations missing data simultaneously. And that 20% number, it's more like a guidepost than a hard and fast rule. But these are sort of things that you ought to be aware of if you're thinking of applying these tools. Okay. So we have our data as site by time matrices and any gaps have been filled. Now what? Our wsyn package requires time series to be demeaned, so we at least need to do that. We may also detrend the data, and this is to minimize the influence of shared long-term trends. This is a pretty standard practice for studies of population spatial synchrony, and even using wavelets, those long-term trends can influence the longest timescales that we can measure. We might also transform the data to improve normality. This can be important for our significance testing approaches. We might also standardize the data or z-score it to equalize the variances among the time series. Whether that's desirable kind of depends on your perspective on synchrony and how unequal the variances of the raw time series are. Normalizing the variances is more consistent with perspectives from population spatial synchrony, which has tended to use matrix like Pearson correlations that do normalize by the variances. And in the package, we provide some standard options for doing this in the clean.dat function. And on this code snippet, I'm using clev for cleaning level equal to 5, meaning the time series for each site are individually detrended, box-cox transformed, and standardized. All right, so from here, let's examine synchrony in beach wrack using the wavelet mean field and wavelet phasor mean field. Dan glossed over this a little bit, but the key difference between these techniques is that the wavelet mean field is sensitive to differences in magnitudes of fluctuation, but the wavelet phasor mean field, the WPMF, can be straightforwardly significance tested, like Dan showed. And so we often use these two things in tandem. We don't have a significance test developed for the wavelet phasor mean field because we don't have a good null hypothesis in mind for what unrelated magnitudes of variability should be. Okay, so we have the wavelet mean field on the left, the phasor mean field on the right. And just to reorient, we're looking at time along the x-axis and time scale on the y. And the heat map colors indicate the degree of synchrony. And then in the wavelet phasor mean field, the black contour indicates statistical significance. And I want to just draw your attention to a few things. On short time scales, less than about a year, we have distinct episodes of synchrony indicated by these splotches of orange and red, those hotter colors. On timescales of about a year, we have much more consistent synchrony represented by this horizontal region, that large, kind of broad band of significance in the wavelet phasor mean field. And then at interannual timescales, periods of about 3 to 5 years, we also have pretty pronounced synchrony. Essentially, the same general features are present in both figures. So hopefully certainly we thought these are some pretty interesting dynamics. Hopefully you think so too. And let's start moving toward investigating what causes them. So earlier, Dan introduced the idea that rather than focusing on each individual timescale or individual wavelet component that the method can resolve, it often makes sense to focus on bands of timescales because this reduces problems with multiple testing, leading to false positives. I also think it helps in synthesizing the results together to make statements about overall patterns. So what sorts of considerations should go into choosing those bands? Sometimes we can do this a priori because we have prior knowledge about a system's dynamics. Excuse me. We have prior knowledge about a system's dynamics and drivers and what timescales they're associated with. There are also some general theoretical considerations, like we've often used 4 years to divide short from long timescales. The justification being that in annual data, a sine wave with period less than 4 years will have negative lag1 correlation, and a sine wave with period greater than 4 years will have positive lag1 autocorrelation. Another valuable way to go about this is to use the data to judge at what timescales there are interesting dynamics to explain. And we tend to think that the more data-driven or context-informed approaches are stronger than that theoretical 4-year threshold. But one way it can be useful is if you are not sure what to go with, or if you need a standard that allows you to compare across multiple systems. So for our kelp wrack study, we considered separately timescale bands of 2 to 8 months, 8 to 16 months. This encompasses annual cycles. I'm going to refer to this a bunch as the annual band, and then 16 to 60 months. The horizontal black lines separate those bands. And in this case, we sort of chose the annual band as something to focus on because we see this consistent high synchrony across it. And the other two timescale bands are basically everything else that the method can resolve. But it kind of fits with the dynamics that we see at the shorter time scales, the 2 to 8 months. Synchrony really isn't consistent, it's more episodic. So there's a difference in dynamics between those bands. Um, and then at the longer time scales greater than 1 year, we see this in kind of range of time scales around 2 years where there's less synchrony, but then greater synchrony at long time scales. So there's sort of a break in the dynamics that corresponds with roughly with the upper limit of that annual 8 to 16 month band. So now that we've picked some timescale bands to focus on, we can run a coherence test. This code snippet shows doing that. Here between wrack on the beach and then our offshore live kelp biomass variable. We're going to use the fast testing algorithm that Dan briefly alluded to earlier, and then the band test function to summarize significance testing across those timescale bands. And then we can plot the magnitude and the phase of the results. So here are those plots. Coherence magnitudes are on the left, phases are on the right. Up toward the top of each panel, you can see summaries for those timescale bands. And you can see that we have significant spatial wavelet coherence between wrack and live kelp at annual and interannual timescales. So this implies that kelp production helps to synchronize beach resource subsidies in the form of wrack across those timescale bands. And on the right, we can see that these relationships are lagged by about a quarter of a cycle, or pi over 2. And the sign of that relationship tells us that kelp production is preceding wrack, as we would expect. One note on the coherence magnitude plot is that dashed red line represents an approximation that's used by the fast testing method. This basically uses a stationarity assumption that helps to speed up the processing, allows us to remove a processing step essentially. And this was developed by Lawrence Shepard. Usually these lines are going to be pretty close. The dashed red line and the solid red line. And so that means it's not really going to affect your conclusions whether you use the fast testing method or another one. But sometimes it's not the case. If these lines aren't close, then that's an indication that the data don't meet that assumption. And you'll want to use the standard slower approach to coherence testing. So in our study, we were interested also in some other drivers that we thought could mediate the relationship between kelp production and wrack. And these are measures of wave action and beach width. Again, waves can damage and dislodge kelp plants, so they influence the supply of detrital material. And then beach width is an index of the depositional environment. A simple way you can think about this is that the wider the beach, the more wrack it can hold. And one thing of note here is that across timescales, a different set of driver variables are coherent with beach wrack. Okay, so the tool that Dan kind of was getting to near the end of his section, but didn't have a chance to discuss in detail, is wavelet linear models. And this is something that we can apply when there may be multiple variables affecting your response variable. And by analogy, you can think of this as sort of correlation is to multiple linear regression as spatial wavelet coherence is to wavelet linear models. So it kind of expands the the framework to help us understand combined effects and interactions between variables. Um, One thing of note, though, is that there are some tools that we have for other kinds of linear models that we don't, or at least don't yet have for wavelet linear models. And one of these is that model selection tools are limited. We don't have AIC, for example. Um, so instead of doing stuff like that, um, we're going to rely on doing sort of more careful variable selection up front, instead of fitting a bunch of models and then trying to discern which one is best. And in practice, careful variable selection generally means focusing on our attention on trying to select and construct variables that have plausible effects on the response variable, just based on logic and prior knowledge of the system. And then testing first for significant bivariate spatial wavelet coherences with the response variable, and for multivariate analyses, only using those that show significant coherences. This is not a perfect system, but kind of right now, this is the best that we have. So code-wise, our structure here is that we're going to provide the data as a list of matrices, and then in our call to WLM, we're going to specify the indices of the response and the predictor variables. A couple slides ago, we saw that the different predictors were coherent at different timescale bands. And so we decided to build a different model for each band. So we're going to focus here on the annual timescale. And here, this is the 8 to 16 month timescale. And here, all three predictors were coherent, so we included all three as predictors in the WLM. But based on this logic, we built different models of wrack at the other timescales that had fewer predictors in it. We can then significance test one or multiple terms by dropping them from the model, and this just means that we replace them with those bootstrap surrogates, and describe whether the model with the real variable explains more synchrony in the response variable than the ones with surrogates. And then in this case, we're dropping all three predictors at once to get a kind of whole model p-value. We already knew that the predictors are coherent with wrack individually, so this is sort of a test of the model as a whole. Looking at some results, we see that the whole model p-value is as small as it can be with only 100 surrogates. We kept that small to speed computation times, but normally that would be at least 10,000 to improve precision, if not 10,000 or more. And then these figures show synchrony as the wavelet mean field for the response variable on the left, and on the right, the predicted synchrony from the model. And you can see visually that this model does a pretty darn good job of recovering the observed synchrony in wrack at annual timescales, which is what the model was built for. So good. It also does a pretty good job at long timescales, but since not all predictors had significant coherences with wrack, and at these longest timescales, we only have a few oscillations and repeated oscillations in the data at those periods, we can really assume that the goodness of this fit is partly due to overfitting. And the model doesn't do a great job at 2 to 8 months. It's really missing a lot of those spikes in synchrony that might be associated with rare events that aren't kind of well explained by this set of predictors. One more thing that we do in the spatial coherence and especially the wave linear model context is investigate the synchrony explained by the model and different components of that model. So by applying this function to the wavelet linear model that we've been focused on, we get output that looks something like this. I constrained this just to the wavelet components in that annual band and left off some columns that are associated with specific pairwise interactions between predictor variables. But looking from left to right, we see the time scale, the synchrony in the response variable at that time scale, the synchrony explained by the model, these cross terms. This is a diagnostic of an assumption of this the synchrony explained in partitioning framework. Lower values of this are better. And then we also have, the model residuals, the contributions of each predictor, assuming that they're acting independently, and then interactions. This column is the sum total of interactions. I'm not showing values of this for specific pairwise combinations of predictors. Right. So interactions can be positive or synergistic, meaning that the actual synchrony is greater than a hypothetical case where all predictors were acting independently. And then interactions can also be negative or antagonistic. This is mostly what you see here. And this means that the actual synchrony is less than this hypothetical where all predictors were acting independently. This can be a bit confusing because it does mean that at times the individual contributions of the predictors on the assumption that they're independent can exceed the amount of synchrony explained if there are negative interaction terms. Okay, so one way that we can sum up that table is to take the average over the timescale band. And so over those annual timescales, we find that the model explains 76% of synchrony, and that the cross terms are about 7% of synchrony explained. Again, that was a sort of a diagnostic test of some of the assumptions of these method. And generally, we've considered less than 10% to be acceptable as a general rule of thumb. All right, so wrapping things up here, hopefully this has helped to make things a little bit more concrete and also clarified some of the important considerations for applying these tools to empirical data, including data treatment, selecting timescales of interest, and model building steps. And then kind of pulling all this together, I hope we've shown that a time and timescale conscious approach to synchrony can reveal overlooked patterns and strengthen inferences into mechanisms, and that the wsyn package provides a nice suite of tools for implementing this perspective. I know I speak for Dan as well in saying that we want people to use these tools and for them to be developed further by us and by others, and that we're willing to make ourselves available to help. I think I have some time to take some questions here, but feel free to also contact us, and our emails are right here. So thanks very much.
Awesome. Thanks, John, and thanks, Dan. That was great. We do have a little bit of time for questions, and so the poll everywhere link is in there if people have questions. I will say while we're waiting, just out of curiosity, so when there is some kind of spatial structure, you suspect some kind of spatial autocorrelation, what's sort of the recommended, like, how do you typically handle that? Would you try to sort of, similar to sort of detrending analysis, would you try to pull out that spatial structure before applying some of these techniques, or what's the sort of general way that you would handle that?
Well, spatial autocorrelation in some sense is what we're studying. So we wouldn't want to remove it. Like synchrony is one thing about spatial autocorrelation, like looking at how these are related to each other across space. We do prevent that from producing from inflating p-values and so on, because all of our surrogates are designed to have the same spatial autocorrelation. So like when you have two data sets in environmental, I'm holding up multiple fingers to represent multiple time series from different locations. So you've got that for an environmental variable, and another one for another set of similar shaped data for biological variable. And you measure their spatial wavelet coherence, which gives you a number, but then you produce surrogate data sets of the environment, and those surrogate data sets are made to have the same temporal and spatial autocorrelation as the original data. Then you recompute the coherence with but they're unrelated to the biology, or they're no more related than you would expect by chance if for variables that had the same spatial and temporal autocorrelation structure. Then you measure the coherence again, and it's that comparison, because of the fact that your surrogates have the same spatial autocorrelation structure and temporal autocorrelation structure. That's where you avoid having that spatial autocorrelation and artifactually inflate your p-values.
Oh, that's excellent. Thank you. We do have a question here from Pull Everywhere. Can you comment on synchronicity for specific events or in systems where you would expect one big pulse of change?
Yeah, I would say that, that is something that is a challenge. This suite of statistics, as I think you probably are getting at, is designed to kind of focus on the consistency of dynamics over time, or the consistency of relationships over time, in large part. Although we, with wavelets can observe changes through time in synchrony, and so yeah, it's challenging. The tools that are in this package are not really set up for detecting specific events.
You can see, like, you'll see an event as a blob in a wavelet phasor mean field or a wavelet mean field, but it might not, if what you think your data show are that, let's say there was a big pulse of rainfall in 1998, and you think that had a big effect in your system. Well, if it just happened the one time and that's what you want to study, probably you want a different set of tools. You'll probably see that pulse, assuming it was synchronous across your locations, in a wavelet mean field. But if you want to establish a relationship between that pulse and the rainfall and an analogous pulse in the biology that you're studying, then these might not be the tools. But if, on the other hand, you think that there's been, like with El Nino, there's been every 3 to 5 years, there's been a pulse of rainfall in the system, and you think that your biology is responding periodically to that pulse, then this is the context that these tools are for.
Great, thank you. An additional question here. Did you compare the wavelet approach to spectral analysis AR models? AR versus wavelet?
AR meaning autoregressive, I guess.
I'm assuming, but
yeah, not sure how to start talking about that. The Fourier methods and autoregressive modeling type methods and the intersection between those two things, they have some overlapping goals, in the sense that you want to decompose signals by timescale or by frequency. And the autoregressive approach to the Fourier analysis would be like a parametric type approach, where you're imagining an autoregressive model of your data and maybe using that to help you construct the Fourier, the power spectrum, let's say, or the spectral characteristics. But the Fourier methods assume stationarity. They don't explicitly decompose or implicitly decompose timescales by how maybe there were changes that took place, like your periodicity changed from one period to another period. And it's just a straight Fourier analysis would average over that and not show you that it was different in the beginning and the end, like some of my earlier examples. Now, you can trick Fourier methods into giving you changes. You could do a windowed Fourier analysis, like a time windowed Fourier analysis, and that'll give you something like a wavelet transform. Basically, the sort of right way to do that is a wavelet transform, but you can certainly do that. So these methods they're both spectral methods. They come from sort of the same place conceptually. But the wavelet methods are designed more to think about changes through time and non-stationarity.
great. Well, thank you. I think we're just a minute over time here, so we're going to go ahead and wrap up, but let's give a big round of applause to Dan and John for the excellent presentation, and thank you both so much.